So good evening, one and all. So I'm Parameshwaran from Center for Ocean Research. Today I'm going to give a lecture on application of fish cell lines. Since there are few students in the group, I'm going to talk about basics of cell culture, and then I will go for the application part. Okay. So the cell culture is the process by which the cells are isolated from the tissues and grown in the controlled conditions. The controlled conditions are nothing but the temperature and the nutrients which we are providing. The nutrients are called media. Okay, so the cells are grown in a controlled conditions of cell culture. So if you are planning to isolate the cells for your research purpose, basically there are three methods you can use to isolate the cells. It may be any of your model organisms. So we used to do it in fish. Okay, so any organism you can isolate. For example, if it is hot, just a minute, I will go from here. So you can go for any of these organs. So there are three different methods. Okay, one is the Explan culture. So Explan culture is, if you are taking the heart, you will cut it into small tiny pieces. Then you will seed it in the growth media. So the cells will start coming out from that explants. The other method is the mechanical disaggregation. So for example, the soft tissues like a brain or liver or the spleen, you can use that syringe. Okay? So using the syringe, you can mechanically force them to get the cells out of the tissues. The other method is using the enzymatic disaggregation method, where you use a pronase, trypsin, or collagenase to digest the tissue, and then you will get the cells directly. So in this both method, this mechanical disaggregation method and enzymatic disaggregation method, you will get the cells directly from the tissues, whereas from the Explan method, you will be seeding the tissues. Okay, so the cells will come out a bit slowly actually. So here in these two methods, you are getting directly cells, and other methods you will get the explants, and from that explants the cells starts coming out slowly. So the cells which comes first is called primary cells. Okay, it's not a homogeneous population; it's a heterogeneous population. So once you started growing these cells continuously, so if you are splitting from one flask to other, it is called passage. Okay, so if you are continuously, if you could able to grow the primary cultures, then if you cross more than 30 doublings, then you can declare as a cell line. Okay, if it is a cell line, it will grow faster, and you can use that for any purpose, any research purpose. Okay, so it will be immortalized spontaneously. Usually whatever cells which I developed previously, it is spontaneously immortalized. We didn't induce any viral for them to grow continuously. So then we need the substrate to grow the cells. Okay, so we use the petri dish or the tissue culture flask with different centimeter square. So in this we can go in both the suspension culture and adherent. So there are two types of cultures. Suspension cultures are the cells which don't adhere on the substrate, whereas adherent cells will go grow by adhering. Okay, so these are all, by growing in this we can get the two-dimensional structure. If you want to grow the cells in three-dimensional, we will use scaffolds. Okay, so these are the basic substrates for the cells to grow. Then the next one is we are providing the nutrients. Okay, so this is the nutrients which has all carbohydrates, vitamins, trace elements, and the inorganic salts and glucose, whatever. So in case of fish, we use Lebowitz L15 mostly. We use that Lebowitz L15 media. So it has all these nutrients actually. So it is also having the phenol red as an indicator. So usually the media comes in a pinkish color. So if the cells utilize the energy, it will becomes acidic actually. It will turn into yellow. So then we can, by visually we can say the cells have we have to change the media. So if it goes more alkaline, then the pH has been increased. So that is the indicator. So for that the phenol red is used, and then the buffers are used to maintain the pH of the cell culture. So then the additional supplements we have to add, that is called the antibiotics and the antibiotics and antimicrobials. So we will use 1% of penicillin, streptomycin, and fungizone. Amphotericin B is also called as fungizone. So to get rid of the contamination, we used to have 1% in any of the cell culture. Then we used to add the fetal bovine serum. This is one of the costliest ingredients in the cell culture media composition. So this is the one which helps the cells to grow. It has more growth factors, so it will help the cells to grow. So other than that, we can also add some supplements like L-glutamine, non-essential amino acids, and then some other growth factors. So if you really want to grow, develop a cell line, you cannot just like that, all the cells cannot grow. So we have to provide a proper environment for them to grow. So for that, we can we can give some additional growth factors like fibroblast growth factor or epidermal growth factor or transforming growth factors. And we can also use the conditioned media. The conditioned media is nothing but the used media from the previous cell culture. So that will have the growth factors to help these cells to grow. So this is the one of the supplement we can provide for the primary cells to get us a cell line. Then also we can use the embryo extract. Actually, for example, if you are using the fish, actually you can use the embryos and you can grind the embryos like 1 ml per 1 embryo, something like that. So that extract, you can add it in as a media supplement. That will also help the cells to grow continuously. Instead of fetal bovine serum, you could also use the fish serum directly. And the other method is like feeder layers. The feeder layers is nothing but growing your primary culture on the top of the established cell line. So these are all the different methods where you can develop a cell line from the primary culture. Excuse me. So you can develop a cell line by using any of this method from the primary culture. You can develop as a cell line. Sorry. So then what I said, the passage actually. So if the cells form in the flask, if the cells forms 70 to 80% confidence, it looks like this. You have some small spaces in between. So if it is 100%, you can see there is no space all with cells. Okay. So if it is 80 to 90, we can go for the subculture. So that is like we use trypsin so that it will chelate the attaching molecules so that we can subculture into 1:2. So once the flask is filled with full of cells, then you can divide it into 1:2. So like that, you can then the 2 flask grows, then you can divide that into 4, something like that. You can subculture. So the main enemy is like the contaminants. Okay, so the bacterial and viral and mycoplasma, these are the basic contaminants in cell culture. And if you are preparing the media by yourselves, you can find metal ions in the water. And also if you are cleaning the vessels, there will be a disinfectants from the detergents you are using. So better you can go for the ready-made pipers or whatever you are using for cell culture. So if before going to your cell culture room, you have to turn on the UV for 30 minutes, then you have to wipe your hands with ethanol, and then you have to go and start your experiment by wiping all the medias, whatever you are using. So this is a basic aseptic technique you have to follow in cell culture. So if you want to get rid of contamination, you have to do the culture in aseptic technique, and as well as use of antibiotics, and also the cell repository that I will talk later. Okay. So this you have to do properly. Actually, what I recommend is better you can go for all ready-made medias. Don't go for the initial. If you are starting your experiments in cell culture, better go for ready-made ones, not by preparing by yourself. So once you have established a cell line like that from the primary culture, if you could be able to develop a cells, then you have to do what type of basic characterization you have to do. So if you are going for publication, then you have to tell about the nature of that cells. Okay. So first one is basically the fetal bovine concentration. So this is I said this is the costliest one. So you have to see which concentration the cells are growing better. So basically from 2.5% to 20% have been used in all the mammalian cells or whatever cell. So you have to study which concentration is working okay with the cells. Then you have to see the proliferation rate using the different temperature. So the incubator, you have to maybe from 22 to 37 degree for mammalian. For fish, actually it will grow maximum 30, 32. That's it. Not 30 to 32 are very rare. So 28 is the optimal temperature for fish cells. So you have to see which concentration, which temperature the cells are going okay. Then the third one is the karyotype actually. So you have to see the karyotype of that organism. So it is maintaining that model number. This is just one of the characteristic feature, but it is not necessary in fish since we are not going for any therapeutic purpose like applications. But anyway, you have to do the characterization saying that this is maintaining no changes in the chromosome, there is no abnormalities. So you have to do karyotypic analysis. Then any cells you are developing, it should have the capacity, it should be able to develop a clone. So if you have to do it in a serial dilution, so from single cell you have to develop a cell line or a cell line. So this capacity should have, if you are saying it is a cell line. So this is one of the characteristic feature you have to do if you have already developed a cell line. Then the phenotype of the cells you can identify by using the immunocytochemistry, using the specific marker for example, fibronectin for fibroblast cells, or pancytokeratin for epithelial. So what type of cell it is, what ZNS5 for osteoblast bone cells. So what type of cell it is, you can confirm by using immunocytochemistry. Then any cells which should have a transfectability, that is, if you are planning to express some protein, so you have to do the transfection ability of these cells. How much percentage of transfectability can be happen by using any of the transfection methods? So this has to be reported. Then the cryopreservation. This is nothing but freezing the cells in minus 196 degrees centigrade. For example, if you're going for vacation, you cannot keep on growing your cells in culture. So you can put it in minus 196. Whenever you're coming back, you can retrieve these cells. So you have to study the retrieval percentage actually. Not every person, if it is established, you can retrieve more than 90% of cells back. But if it is in a primary culture or in between the transformation between primary to cell line, then the revival rate will be very low actually. You will lose the cells actually. You cannot revive the cells, everything will die. So these are all the basic characterization you have to do once you have the cell line. So then you can go for publication. But this is just about telling this what is the nature of the cells. Then if you are planning to work for the application, then I'm coming to the application. So these are all some of the applications from Fisher lines, this is from my research actually. So basically Fisher lines are used for virology, okay? But now not recently, maybe 2 decades, actually it is used in stem cell research, also in bone research. So these are the type of research applications I will explain one by one. So first we can go for stem cell research, okay? So this is a project we recently got, not recently, 4 years back we got from DST. So the main idea of this project is to develop the stem cells from the blastula stage of zebrafish, okay? So for that we will use the blastula stage. From that stage, the blastula, we will isolate the cells from any of the methods which I told you back beginning. So that cells, we will be stably transfecting a gene which always express red color, that is red fluorescent protein. So then if you have this red fluorescent protein expressing cells, then we will inject them into the blastula again to see these cells are having the in vitro potential, that is whether the cells are taking part in the organogenesis of this particular fish, okay? So this is the study actually. Then we are also seeing what are the type of lineages these cells can able to differentiate. So this is one of the project we got, and just I'm explaining, I'm going to tell about the results of that, okay? So this is a zebrafish developmental stage. So this is the 250. This is the blastula stage. So from the blastula stage to this 50% epibole stage, you can get the stem cells, that is pluripotent stem cells. We usually get the cells from this blastula stage. That is what all recommended. But we are not able to succeed in zebrafish, so we have used the other model, that is sea bream. So this is the blastula stage. So from the blastula stage, we have isolated the cells using the enzymatic disaggregation method. So these stem cells will grow as a colony, actually, always it grows in a colony. Then also stem cells will express high alkaline phosphatase activity. Then these cells are positive for OCT4. This is a transcription factor only expressed in undifferentiated cells. Then SSEA1 is also an undifferentiated marker. Then this is called embryonic body actually. This is a 3-dimensional structure aggregates actually, which is grown in suspension. Only the stem cells is having these features actually. So these are the characteristic features only the stem cells will display actually. So we found that the cells also show all the mammalian in vitro differentiation markers actually, like OCT4, alkaline phosphatase activity, and embryonic body formation. Then we triggered these cells towards a bone phenotype. Since we worked in a bone, so just we want to see whether these cells can differentiate into bone. So the origin is from the blastula, okay? So whether these cells can be differentiated into bone, we have checked with the 3 weeks of treatment with chemicals like vitamin C, beta-glucophosphate, and ascorbic acid. This is the cocktail which will induce the normal cells to bone actually. So this is the phosphate and calcium. These are the bone is formed with calcium and phosphate. So these are the markers which confirm these cells are osteoblastic phenotype. Then we also triggered the cells towards the cartilage lineage actually. That has been confirmed with Alcyon blue technique. This is one of the stain for cartilage. Then we have confirmed with various immunocytochemistry protein specific for both the osteoblast and the chondroblast. So we found after, this is the time one week, this is the time 0, those are time week, you can see some expression, and this is the time 3. So you can see more protein expression of the osteopontin, osteonectin, and bone alkaline phosphatase on the 3rd week. So similarly, it's a collagen 2, 10, and a chondroitin sulfate proteograph. This is the marker for cartilage. So these cells can differentiate into both cartilage and osteoblast. Then we have confirmed this OCT4, this is undifferentiated marker in different various passages. That is in 14 also it expressed, and in passage 28 also it expressed. Then collagen 2 is a gene from bone, so this is also expressed by real-time PCR. So then other than that, these cells spontaneously differentiate into cardiomyocytes actually. Just a minute, I think the video is not playing. So you can see there's a pumping movement of these cells and here. So in the culture, these cells are spontaneously differentiated into cardiomyocytes. If you know that if you are triggering them towards cardiomyocytes, you have some cocktails, so you can also trigger them towards heart cells. So this happens in spontaneously. Actually, this is another phase of the same flash. So you can see here. Then we have triggered these cells to with retinoic acid, which usually triggers towards neuronal lineage. So we have confirmed that with these are the neuronal cells after treating with retinoic acid. So this is melanocyte pigment cells. Then this is muscular cells. So these cells can able to differentiate into many lineage when we are triggering them with retinoic acid. This is a benzene staining for blood cells actually. This is osteoclast, is the another bone cell type. So this is also positive. So this is the one of the cathepsin K. This is the protein which expresses in osteoclastic cells. So this is also positive for cathepsin K. So from this experiment, we can tell that the cells can able to differentiate into osteoblasts, chondroblasts, and neuronal and melanocytes and cardiomyocytes. So it can be differentiated into any type of lineages. So this shows this have the in vitro differentiation potential. So we have to see then the in vivo differentiation potential, whether these cells can able to play a role in organogenesis. For that, we just develop a stable clones actually. So this is the embryonic stem cells. We have transfected them with the DS red N1 actually. It's a plasmid which express red fluorescent protein. So we have developed a stable clones which always express the red fluorescent protein. So we just take these cells and then we inject it in the blastula stage of the zebrafish. Actually, we have started the story in a sea bream, but the sea bream eggs are very tough. We cannot do the micro injection. So we just check this, say in a zebrafish, actually, zebrafish embryos. So we could see the cells can take part in organogenesis. So it's a trunk region. Actually, it's a vertebra. So it's a gut actually. You can see there's a lining of cells in vertebra and also in a trunk region. So this is the 30-hour post micro injection. You can see different different region actually, where you can see the expression of these cells actually. This is 1 to 120 hours post micro injection. So these are all these cells have the capacity also in vivo actually. So then we start doing the basic characterization. So the transfectability. So we use different methods: calcium phosphate, fugine, and nucleofection. The nucleofection method is like giving electrical pulse so that the plasmid can go easily. So by using this nucleofection, we could able to transfect more than 50% of cells using the L4. It's a buffer with that equipment itself, you will get different buffers. So using the L4, we could able to transfect more than 50%. So it's very nice transfectability. Then the karyotype, we found that the cells are maintaining the 48, the model chromosome number of that C beam. Then the proliferation rate we used to check for the FBS concentration. So these cells can easily grow in a 5% serum, comparatively for 20. Basically, stem cells don't need much of the serum, so it will go easily in the 5%. So in conclusion, this is the cell line we developed, that it has both in vivo and in vitro properties, and we named this cell line as ESA1. Now we are working in cobia ES cells. Actually, we are trying to develop cells from cobia. So this is the one cell line we developed. So this is the stem cell application. So here we use the gene cut is a DS red. So you can also do, if you want to make the cell fish to grow fast, you can clone the growth hormone gene or whatever. So you can play accordingly. So you, whatever gene or whatever interest, you can if you have a stem cells, you can do anything actually. That's what I would like to say. So then how you can use the cells for a bone research. So okay. So zebrafish and guppy models are widely used to study more bone-related diseases such as osteogenesis imperfecta, Karyotype syndrome, idiopathic scoliosis, undersponding. So these are all the bone diseases actually. So you may saw this kind of curved vertebra actually. So this is idiopathic scoliosis actually. So this is this kind of skeletal abnormalities you could see in the formed fishes. So that is having an effect in aquaculture industry. So in order to study that, as well as we need one fish cell line model for studying human diseases. So we plan to develop a cell lines from bone actually. So for that we use the experimental model zebrafish. So from that we took the calcified tissues like vertebra and jaw. So from that we have started developing a primary culture. So we have used both the Explan method. Explan, this is a small piece of bone actually, vertebra. So you could see the cells are coming out of this piece of vertebra. So we did both the method actually, that enzymatic method as well as the Explan method. So we got two cell lines actually. One is we named as ZFB1 and another one is ZFB2, but we characterized only ZFB1. So like this, these are all the different passages. So cells are showing the same phenotype. Again, the fetal bovine serum concentration. So we could find 15% is suitable for the cells to grow. Then this zebrafish is 50, model number of chromosome is 50, but we could find 78 actually. This is some abnormalities happen in the chromosome. So that is not a problem actually. We are not going for any medical application or whatever. Then the nuclear fraction, we found the transfectability, we found again with the nuclear fraction, we could see more than 70% cells are transfectable. So if you want to produce any recombinant protein, it's easily transfectable and we can produce anything. So these cells are really high percentage of transfectability. Then since this is from the bone, we have to know whether this is really bone. So then we have triggered the cells towards bone with the same cocktail, which I previously told you. So these cells are positive for this phosphate and calcium. This is a 1% alizarin. Then alkaline phosphatase is one of the marker for bone, and this is highly expressed in normal in control as well as in treated, and alcyan blue staining for cartilage. So these are some protein markers for chondroblastic, osteoblastic. So these are all for 3 weeks of treatment. So then from real-time PCR, we could see the gene specific for bone and cartilage is expressed. So this RUNX2 Nostrix or transcription factor, which will express during the late differentiation process, that is 3rd week. So this alkaline phosphatase is the initially it will express. So you can see up to 1st week it is expressed. And this is SOX9 is a chondroblast marker, so this can be seen up to 3 weeks. Then SPP1 is spark, and collagen 10, collagen 2 is chondroblast, and these are, so you can see all highly expressed during the 3rd week. So this is the cell line. We named it as ZFB1. It has it can be differentiated into chondroblast and osteoblast. So this is the tool actually you have from bone. So you can study anything related to bone. If you have this cell line, you can work in the lab itself. You don't have to go for animal something. Then the third application I would like to say is the regeneration study. Not very few groups are working in this aspect actually, regeneration. So if you cut the caudal fin or heart, or even the frog is amphibia, and actually the fishes and amphibia is having this unique property, and even the starfish. If you cut, it will regenerate actually. So if it is a zebrafish, if you cut the caudal fin, it will regenerate in 15 days actually, entire fin actually. The reason for that is there is a specific cell type called the blastema. So that is the responsible for the entire regeneration of the amputated fin actually. So for that, we plan to develop a cell line from the blastula. So for that, we have designed a protocol. For that, we have used 3 different species actually: gilded sea bream, western mosquito fish, and zebrafish. So this is the thing. So this is the fin actually. This you will say this is a bifurcation. Before the bifurcation, the 3 segments before we will amputate. So the 48 hours after amputation, you could find a group of cells called blastema. So these are all the cells which is having the high differentiation potential, so which is responsible for the entire formation of the missing fin actually. So we took these cells, this blastema, after 48 hours of amputation, this explant. Then this is the basic protocol. We use this collagenase actually, enzymatic disaggregation. So we took from 15 fishes or more. Then is a protocol actually. We finally got the cells. So from these three species, we could find the cells like this: zebrafish, gilded silverworm, and a masterfish. Then we just checked for the FBS again. So we could find 15% is okay, 5% is okay for all the cells. Then if you, these cells are very difficult to grow, so we provided the extracellular matrix, that is a coating actually in the flask. So there is this extracellular matrix will help the cells to adhere and it will trigger the growth actually. So for that we have used fibronectin, laminin, polyl-lysine, and gelatin. These are all the extracellular matrix, and one is non-coated. What we found is without any coating, the cells are okay. It don't like the extracellular matrix. So we grown the cells in without any ECM. So then we just focused only on zebrafish. So this is how these cells grow. This is a colony-like nature actually. Only embryonic stem cells will grow like this. So then in culture, these cells can differentiate into spontaneously differentiate into many type of cells like neuronal. So you can see this is the normal phenotype, and this you can see the neuronal cells in culture. Then we also induce the cells to see whether this can go to neuronal with retinoic acid induction. So we could see the neuronal differentiation of these cells. Then we confirmed with some specific markers for bone. This is ZNS5 also bone. This is for neuronal. This is an undifferentiated marker, and this is a proliferation marker, and then this is a negative. So you could see this, even the cells are not highly proliferative. You can see this out of these 5 cells, this cells is not positive. So we have designed a protocol to isolate these cells, but these cells can able to subculture for 1 or 2, maximum 2-3 passages, then it will all disappear. So we are not able to grow continuously. This is from zebrafish what I'm talking. So this is the protocol we designed, and we also published this protocol. So we need a cell line from this. So we have we have used this CBRIM as a model. So the same thing we have did with this. So we have isolated these cells, and we have already characterized, and we could see the differentiation potential. But now we are just working on this, getting the stable clones, so that our plan is to re-inject this in the developing fin to see whether these cells are again taking part in the regeneration process. So this is the step we are working now. And also, since these cells have more differentiation capacity, so we also planning to do it in a microinjecting in blastula to see whether these cells are taking part in organogenesis also. So this is the part we are currently working on. So this is the thing, okay. So it is a 0th hour we are amputating. So this is a 48th hour. You could see the small layer of this is a cell actually, cell mass actually. This is called plasma. Then this is 72 hours. So day 7, day 8. So on day 14, you could see the entire fin formation. So this is the amputated line. So you could see the entire fin formation. This is from CBRIM actually. So we took that 48-hour amputated blastema. So this is the one. So since this fish fin is bigger, just we took the half of the. This is the only piece we use for this cell culture. So this is ion magnification. So from that you could get the cells out of that. So again, then once we get the cells we have subculture more than 100 passages, and this is the morphology which is showing the phenotype. Then again we have checked for the growth of cell proliferation, and then like a transfection. Then we have just checked for a bone differentiation potential. Then we have confirmed with various protein markers for bone and bone actually, specifically bone. And these cells also able to go differentiate into osteoclast. This is again the immunocytochemistry for osteoclast. Then this is the osteoclast actually again. So this is a resorption pit actually. It is an osteogenic disc. It is coated with phosphate and calcium. So if you are seeding these osteoclast cells, it will do the resorption process actually. You can you see this, it eats all the calcium and phosphate. It's a different region of the disc actually. So these cells can able to differentiate into osteoclast. So Then the gene specific for cartilage and osteoblast, we could see the expression. So this is the thing, 40th day amputation, we could see entire fin formation. This is alizarin staining. Actually, in the 6th day, we could find that bone formation, and 9th day, you can see all the bone, the missed bone formation. And here it shows the osteoblast formation at 48 hours amputation. So since we are working in having a stable clone from blastema, but we already have one clone from that bone cells, actually ZFB2. We didn't characterize, but we have developed a clone from this. But it is, these cells are originated from bone. Just we have checked this in regenerating fin of sea breams to see whether this is taking part in regeneration. But since this is a fish is entirely different species, as well as this is not stem cell, just we have standardized the microinjection protocol whether these cells can be able to inject in the regenerating fin. So we have injectors. You could see the cells, actually the cells are actually some in the fluorescent microscopy. You could see the injected cells after 18 hours of microinjection. The cells are growing actually in so now we are in now we are actually, so this is the cells we have named it as BSA2. So it is having multipotency capacity from the blastema. So we are now currently working on the developing the stable reporter lines, actually. So this is the one of the application in regeneration. Not many people are working, very few groups in the world are working. So this is a nice area if you are planning for research in regeneration. Then next one is the virology. So this is a cell line maybe 10 to 15 years back we have developed. So basic characterization again. So then we have treated the cells with a different fish virus like betanodovirus and Marine Bio virus. So this is the normal culture. So after infection, you could see this one single cell having multiple vacculations. So this is a nodovirus. We used to say this is the typical characterization of that infection, that nodovirus infection. You could see multiple vacculations. These are all the lengthy things are stellate formation. We used to say this is also the Marine Bio virus. So these cells, with the developed cells, can able what I'm saying is these cells are able to we can replicate the virus. This you can propagate the virus using these cells, actually. So you can get you can see this titer is almost doubling actually after 7 days, after 3 days for Marine Bio and nodovirus maybe it's kind of 6th day. So you can propagate the virus for working on attenuated vaccine or whatever. Then this also positive for in PCR. So this is the TEM analysis. So this is a single cell actually having a multiple vacculation. You could see the single cell having multiple vacculation. Then these are all the nodovirus. Again, recently we have developed this mosquito fish cell line, and this is not susceptible for nodovirus, but there is only one report saying that this is susceptible. So we developed this and we just characterized the basic characterization. Then we have seen again this nodovirus is infecting. This is a normal one. So this is again infecting. So you can see multiple vacculation. So we have used 2 strains of nodovirus. Both are replicating in this developed cell line, and this is positive for a PCR as well. So these cells, this fish is not really. What happened is this is the cell. The color of the fish is changing after a few days. Then the descaling happening, and so the scales, all the scales are falling down after injection actually. So the mortality we could see maximum 20% after 25 days. Actually, it's not really highly mortality, but this virus is not specific for this species. But just we tried, but cytopathic effect is happening, but it's not highly infectious for this mosquito fish. So these are the some same analysis. So this is the normal images, and this you can see budding virions can be seen in the infected ones. So this is basically you can use for virology. Any fish virus, if you want to propagate or if you want to do attenuate vaccine, you can go for this virology. You can use this fish cells. Then there's the toxicology. This virology and toxicology, basically everyone is using cell lines. The other application which I said, I think it is a kind of new maybe for you, for someone, for many, I hope. So toxicology, actually you can use nanoparticles or whatever if you want to take the drugs or cytotoxicity, you can use. Just this is only one slide I'm showing. This is the effluents from the chemical and tannery effluents that we have just taken. So before filtration, any factories, they will filter the discharge before, filter whatever the waste they will filter, and they will allow them in the gutter or the river actually. So we have collected the before one and after one, we have checked, but we could see only the before one is having the high effect. This is a normal one, but for using the three effluents, we could see the cells are dead. More cells are dead using this other the prior treatment effluents actually. So after treatment, there is no effect. We could not see anything. So this is just I'm saying. So you can use these cells for toxicology application. For that purpose, I'm just showing this slide. So very recently, this is the latest, and actually I would say the cellular agriculture. So this is I want to say this application. So this is it is expected that in 2015 there is no enough seafood or even pork or meat for the growing population. So for that, this concept of cellular agriculture has arrived actually. So by using in cellular agriculture, you can produce animal foods from cell culture using a combination of biotechnology, tissue engineering, and molecular biology. So it's a very recent innovation. So if you are developing it is from seafood, the name for that is cell-based seafood production. Okay, so you can generate seafood without the need of animal. So you don't have to have the animal. So you can just prepare the meat from the lab. So we recently got one project from this aspect actually from Singapore, and we are working on that. So the main idea is to have so first you have to see the commercially valuable fish. Actually, you have to develop. If the price of fish is too much, then that is nicer actually. So if you're going for tilapia and all, it's very cheaper price, so you don't have to. So you have to see the commercially valuable fish. Then from three different ways you can get the muscle cells actually. So one from the muscle, you can directly take the cells from the muscle and you can grow the muscle cells, or you can take from the stem cells, or you can go for iPSC, that region, induced pluripotent stem cells. You just cut the caudal fin, you get the fibroblast cells out of that, then you reprogram that cells into myoblast. So from the myoblast, you can you can go for the next step actually. So by doing three different types, and also there are many types, so you can get the muscle cells. So once you have the muscle cells, then the second aspect for this is you have to get rid of the serum from the media actually, because since it is if you are going for bulk production biomass, then the serum is the highly more costly actually. So you have to get rid of the serum, and other animal product in the fish is not acceptable. So you have to get rid of serum. So for that, we have to find the alternate serum actually. It's not serum, you can use plant source or marine source or anything. So that is the second aspect. The first aspect is developing a muscular cell line. Second aspect is to develop a media without fetal bovine serum. Then third aspect, it will comes for a scaffold actually, scaffold or a biofermata. So you have to go, once you have the cells, you have to grow it in a three-dimensional structure. So for that, you will coming to the third aspect, that is the three-dimensional, the scaffold development. And then you are going to grow them in a biomass, say coming as a biofermata. Then the last one is it goes for the chef for flavor and taste actually. So finally you get the product something like this. So it can be grown in lab actually. You don't have to go like for the growing population. Definitely this field is highly promising, and actually I would say more than 50+ companies already started working in this area. And maybe around 10 companies are working in developing seafood, actually, cultivated seafood, and many are chicken, meat, pork, and all. I think one Delhi-based company is working in chicken as well. So this is one of the promising area. So using fish cell lines. So these are all some of the applications of fish cell lines. So if you want to know what are the cell lines available, you can go for this Cellosaurus link. So you can see all the cell lines available so far in this world actually. So if you want to work on any specific fish cell lines, you can access in these two places. One is National Repository of Fish Cell in Lucknow, and Aquatic Animal Laboratory in Vellore, Tamil Nadu. So here you can get your, if you are planning to work in fish cells, you can get it from these two places. Then the cell lines which I have presented can be accessed in same in Visharam as well as in the bone cell line and stem cell line and all you can access in Center for Marine Science, Portugal. So this is my talk actually. So I thank the management for the constant support in research activities. And I also thank our director research madam and our HOD and also my colleagues. So I also thank my funding agencies like FCT and the DST. Thank you. If you have any questions, I'm happy to answer.
Thank you, sir. Thank you so much for your wonderful presentation.
Thank you.
Participants, if you have any questions or queries regarding the topic, you can unmute and can interact with the speaker, or else you can put your questions in the chat box.
Okay, if there is no questions, shall we move to the next session? Thank you, Dr. Parameshwaran. Actually, it's really wonderful. Whenever I am listening to your talk, I feel it is a new area for me.
Thank you.
And I think some question is in the chat box.
They have congratulated the work, sir.
And one more thing is from Ramya.
Yeah, from Ramya, one more question is there.
Yes, yeah, you can tell me. Yes, plant-based ingredients. Yes, they are doing that. Yes, plant-based ingredients they're doing. It's called protein hydrolysates actually. So they're doing actually.
Thank you, Dr. Parameshwaran.
Thank you, sir.
Thank you so much, sir.
Thank you.
So